right, so this is chapter 3. I know we kind of skipped around a little bit, but I was trying to, again, coordinate this with our labs. So we're going to break this up into parts. Part 1 is organic molecules. So there are many molecules found in cells, and they are composed of carbon. Carbon is our very important element. Remember, it has 4 valence electrons, so it can bind to all kinds of other carbon atoms and other elements. Carbon-based molecules are called organic compounds. So generally, when we talk about an organic compound, we are talking about a compound that is composed of carbon and hydrogen. CO2 has carbon, but it does not have hydrogen, which is why we generally say carbon plus hydrogen would give it that designation of organic. So we don't have to say except for carbon dioxide and carbon monoxide. The organic molecules that we are going to be focusing on this semester are these 4 right here, and we're going to do a lab on those 4: carbs, proteins, lipids, and nucleic acids. So again, by sharing electrons, carbon can bond up to 4 other atoms. So we can see that here. 1, 2, 3, 4. These 3 models are different types of models we would use in chemistry. They can form either straight chains, branches. They can even form some rings. We'll talk about sugars being rings when we get to carbohydrates. This is the simplest organic compound. It is methane, which is CH4, 4 hydrogens bound to a single carbon atom. A carbon skeleton is a chain of carbon atoms that can be branched or unbranched. We also have this thing called isomers. Isomers are compounds with the same formula, but different structural arrangements. So notice how they are both C4H10, but they are organized differently. So butane is a straight chain. Isobutane is a straight chain, but we have this carbon branching off of it. And so we have to designate which type of structure we have here, whether it's a straight chain butane or it's branched like isobutane. Notice they both have the same number of carbons and hydrogen, so their formulas are the same. They're just organized differently. So that's what an isomer is. So do make sure you are familiar with that definition. An organic compound has unique properties that depend upon the size and shape of the molecule, and we have these groups of atoms called functional groups. A functional group generally affects the biological molecule's function in a characteristic way. Most of our functional groups are going to make them hydrophilic, which means they like water. They will be soluble or will dissolve in water. Here is a table of some of the functional groups. You will find that in your textbook. We have what's called a hydroxyl group, which is an OH, a carbonyl group. There are a couple different carbonyl groups. You can have a carbonyl group in the middle of your molecule. That's called a ketone, or at the end. So this is the last carbon, and that is called an aldehyde. We also have a carboxyl group, which is a C with 2 oxygens here and an OH, right? That would be a hydroxyl group attached to it. So basically, it's kind of a combination of a hydroxyl and a carbonyl. So we get a carboxyl. An amino group, which we have NH2. Phosphate is a bunch. We use phosphates a lot in different molecules we'll talk about in here, which is a phosphorus atom surrounded by oxygens, and our methyl group. So we can see in this example, we have a male and female lion. Both of them have been identified with their gender when they were developed based on these right here, these sex hormones. We have testosterone and we have estrogen. Notice the only thing different basically are these functional groups. And so that is the biggest difference between those 2 molecules. There are 4 classes of molecules important to organisms. We're going to talk about each of these in a different part for this chapter. So we will talk about our carbs, proteins, lipids, and nucleic acids. The 4 classes of biological macromolecules contain very large molecules, which is why I referred to them as macro meaning big. They are also called polymers. Poly means many. Those large molecules are generally made from what we call monomers. Monomers are smaller blocks. So we're going to put those monomers together to get these polymers. So if we start with these monomer groups, we're going to put them together. We're going to bond them together, right? So these would be our monomers, and this would be our polymer. So it's made up of many units. So how do we do that? So there are 2 different types of reactions we're going to talk about. We have what is called a dehydration. So dehydration means it pulls out water. So we're going to take those 2 end caps there, because remember, everybody's balanced based on valence electrons. And we're going to have this polymer, this small polymer that's made up of 1, 2, 3 monomers. And we're going to attach number 4 here. When we do that, we have to pull out a water molecule so we can form that new covalent bond. So this is often called dehydration, because we're dehydrating or pulling out water. It is also referred to as a condensation reaction, because like condensation on your glass, like it leaves water, because we're pulling out and forming water with that chemical reaction. So here's a picture of that. There's our short polymer. We're going to pull out that water, and now we have a longer polymer. We also have the opposite. So we're not always building up. So again, that's also called an anabolic reaction. Anabolic means to build up. We also have what's called catabolic, or to break down. And so when we talk about this, we are basically doing the opposite. We're going to have to put water in. We're going to split the water, right? So then we split it. So we put our end caps back on, and we're going to pull off that monomer. So this is like important in digestion. When you're breaking down your proteins, you're going to break them down into the smaller monomers, which is amino acids. So here's our polymer. We're going to add our water, split it up, put our end caps, I always call that my end caps, back on. And now we have broken that down. That is, again, that would be catabolic versus anabolic. Okay. Now, one of the last things this unit talks about is enzymes. All of these reactions, whether they are anabolic, catabolic, dehydration reaction, hydrolysis reaction, those are all sped up by an enzyme. Enzymes speed up the rate of a chemical reaction. So we see an enzyme here, and we talked about this earlier. So we have our enzyme, we have our substrate. Our substrate here is a 2-sugar molecule. We're going to add our end caps. So right here is our water. We're putting our end caps on. So this would be hydrolysis reaction. This is speeding this up, and it is breaking it down into their monomers. So a small polymer into the 2 monomers. We have to use that water through that hydrolysis reaction. And then we talk about, again, what I've been talking about here all along, monomers versus polymers. So a cell makes a large number of polymers from a small group of monomers. For example, proteins have 20 different amino acids. DNA is made up of 4 nucleotides. So this would be 20 monomers, 20 different monomers. DNA makes itself up. It's composed of 4 different monomers. We're going to talk about carbohydrates, only have 1 type of monomer. All right, so you need to know the difference between the monomer and the polymer. And we'll be talking about that in many different places, and you'll see I've had many different charts with those in there. All right.